Hello everyone, I am Dr. S. V. Rankham, Assistant Professor, Department of Zoology, late Rameshwar Pudkar ACS College, Sonpet, District Parvani. Dear friends, today we are going to see the syllabus on paper number 7 for B.Sc second year. We know paper number 7 is a biochemistry. What is biochemistry? Biochemistry is the branch of science that explores the chemical processes within and related to living organisms. It is a laboratory based science that brings together biology and chemistry by using chemical knowledge and techniques. Biochemists can understand and solve biological problems. This syllabus is divided into 4 units. Then unit first for 12 marks. Between them, first point is a biomolecules. Biomolecule, also called biological molecule, any of numerous substances that are produced by cells and living organisms. Biomolecules have a wide range of sizes and structures and perform a vast array of functions. The four major types of biomolecules are carbohydrates, lipids, nucleic acids, and proteins. In this first point, we see classification, structure, and properties of carbohydrates. We know carbohydrates or carbs are sugars and polymers of sugars. The next point is a classification, structure and properties of proteins. We know proteins are polymers of amino acids. Then classification, structure and properties of lipids. Lipids are non-polar molecules. Then unit second, water. We know water is essential for life. Water covers 75% area of earth. Water is a solvent of life. Water has many special properties. Between them, in this unit second, we will see electrochemical properties of water, pH and correlative properties. Then next point is enzymes. We know enzymes are proteins that act as a biological catalysts. Catalysts accelerate chemical reactions, and this first point is a nomenclature and classification mechanism of enzyme action. ES complex formation, lock and key model, induced fit theory. Then factors affecting enzyme activity like temperature, pH, concentration of enzymes, concentration of substrate. Then unit number third, metabolism is a term that is used to describe all chemical reactions involved in maintaining the living state of the cells and the organism. Metabolism can be divided. Metabolism can be conveniently divided into two categories. Number one is a catabolism and number two is a anabolism. Catabolism plus anabolism is equal to metabolism. In this point, we will see carbohydrate metabolism. We know glycolysis or EMP pathway. This EMP pathway allows the metabolic use of glucose to generate ATP, NADH, and several biosynthetic precursors such as 3-phosphoglycerate or pyruvate. Precursors such as 3-phosphoglycerate or pyruvate. The next point is a glycogenesis. We know glycogenesis is the process of glycogen synthesis in which glucose molecules are added to chains of glycogen for storage. The next point is a glycogenolysis. Glycogenolysis is the breakdown of the molecule glycogen into glucose, a simple sugar that the body uses to produce energy. Then glyconeogenesis. We know glyconeogenesis is the formation of glycogen in the liver form from non-carbohydrate sources such as amino acids and lactate. Then citric acid cycles, which is also called as a Krebs cycles. The citric acid cycle, also known as a TCA cycle or Krebs cycle, is a series of chemical reactions used by all aerobic organisms to release stored energy through the oxidation of acetylcholine derived from the carbohydrates, fats, and proteins. Then pentose phosphate pathway. The pentose phosphate pathway is also called the phosphogluconate pathway, and the hexose monophosphate shunt is a metabolic pathway parallel to glycolysis. It generates NADPH and pentoses, it, as well as ribose 5-phosphate, a precursors for the synthesis of nucleotides. Then unit 4. In this unit 4, lipid metabolism. The S oxidation or beta oxidation pathway in biochemistry and metabolism. Beta oxidation is the catabolic process by which fatty acid molecules are broken down in the cytosol in prokaryotes and in the mitochondria in eukaryotes to the generate acetyl CoA, which enters the citric acid cycle, and NADH and FADH2, which are coenzymes used in the electron transport chain. Then ketosis. Ketosis is a process that happens when your body doesn't have enough carbohydrates to burn for energy. Instead, it burns fat and makes things called ketones, which it can use for fuel. Then, ketogenesis. We know ketogenesis is the biochemical process through which organisms produce ketone bodies through breakdown of fatty acids and ketogenic amino acids. This process supplies energy under circumstances such as a fasting or caloric restriction to certain organs, particularly the brain, heart, and skeletal muscle. Then ketolysis. Ketolysis is the breakdown of ketones. The next point is a protein metabolism. In this point, we will see transamination, deamination, and decarboxylation reactions of amino acids. Then disposal of nitrogenous waste, Krebs, Krebs Henseleit urea cycle or ornithine cycle. This is our syllabus for biochemistry in B.Sc second year.